Hello and good day. My name is Kaitlyn Flynn, and today I shall be presenting some of the results from my MSc project that was functioning under the Deciphering Developmental Disorders in Africa project. The title for this talk is Evaluating Whole Exome Sequencing on the Thermo Fisher Torrent S5 as a Potential Tool for Diagnosing Developmental Disorders. Developmental disorders occur due to abnormal or delayed physical and mental development. And though there are multiple causes of DD, it is thought that 25 to 50% of cases have a genetic etiology, yet majority of our patients remain undiagnosed. Whole exome sequencing is the recommended first-line genetic test for diagnosing these heterogeneous disorders, due to the high diagnostic rate in comparison to the current gold standard of chromosomal microarray. However, this test is not routinely offered to state patients in South Africa. But the availability of a Thermo Fisher Torrent S5 NGS platform within our department created a unique opportunity to investigate the feasibility, diagnostic rates, and clinical utility of implementing this test within the local context. The overall aim of this project was to implement a whole exome sequencing workflow and evaluate the financial and computational costs involved in this technique. Ethical clearance was received from the WITS Human Research Ethical Committee. We then performed whole exome sequencing on 7 DD patient samples and 1 sequencing control. Data analysis was performed across 3 investigative platforms that were later compared. We performed Sanger sequencing for variant validation and to determine the mode of inheritance of prioritized variants, and lastly performed a cost and diagnostic yield analysis. For the sake of this presentation, I shall be focusing on the results of the whole exome sequencing and the associated diagnoses identified. In this project, we identified possible putative disease-causing mutations in 6 of the 7 patients and were able to validate the variants in 5 of the patients. This investigation identified a patient with Takanushi-Kozaki syndrome, which to our knowledge is the first reported case in Africa, and this was seen in patient 1. We also identified 2 cases of mandibulofacial dysostosis with microcephaly in patients 3 and 7, followed by congenital muscular dystrophy and mental retardation type B2 in patients 4 and 5 who are fraternal twins carrying the same compound heterozygous mutations. Through this project, we achieved and refined a whole exome sequencing workflow for the investigation of DD in South African patients. We obtained a diagnostic rate of 70%, which is much higher than the global diagnostic rate of 40%. However, our samples were enriched with well-defined clinical phenotypes which better led our investigation. Should the test be implemented in the local setting, we would expect the diagnostic rate to drop and match that of the global statistic. Overall, we concluded that whole exome sequencing would be a highly beneficial tool in the investigation of DD within our department. Thank you.